All right, welcome back. In our last explainer, we really dug into the basics of monosaccharides, those simple sugars. Today, we're going to build on that foundation. We're linking those simple sugars together into complex chains that do so much more than just give you a quick energy boost.
You know, when I say carbs, what pops into your head? Probably fuel, right? Bread, pasta, potatoes, stuff that gives you energy. And you're not wrong. But honestly, that's just a tiny piece of a much bigger puzzle. It's kind of like saying letters are only for writing shopping lists.
What if I told you these exact same molecules are at the very heart of some of biology's most complex systems? They act as structural scaffolds, as communication signals, and get this, even as the personal ID tags on the surface of your very own cells. So how on earth do they pull all this off?
Well, that's what we're here to find out. First, we'll get to know the oligosaccharide family and their most famous members, the disaccharides. Then, we're going to scale up to the real giants, the polysaccharides, and see how some are built for storage while others are built for structure. And finally, we'll uncover one of their most amazing jobs, acting as the identity markers that literally determine your blood type. Okay, let's dive right in. We're going to start with the middleweights of the carbohydrate world. Oligosaccharides are the perfect bridge between those simple sugars we already know and the massive polymers we'll get to in a bit.
The name oligo actually means a few, which is perfect because that's exactly what they are. Short chains with anywhere from 2 to 10 simple sugar units all linked together. And just like their smaller cousins, they're usually sweet and they dissolve in water. But their real magic is how they can be combined in super specific ways to create all sorts of unique structures.
Now, the most common and familiar oligosaccharides are definitely the disaccharides. As you can probably guess from the di prefix, they're made of exactly 2 sugar units. Let's meet the big 3.
First up, we've got sucrose. You probably just call it table sugar. It's what you get when a glucose molecule links up with a fructose molecule. Now, the bond that joins them is super specific, a beta-1,2 glycosidic bond. And here's the key. This bond ties up the most reactive part of both sugars, their anomeric carbons. By locking them both up, there are no reactive ends left hanging. This is precisely why sucrose is known as a non-reducing sugar. It's chemically stable and doesn't really want to react with other things. Next, we have maltose, or malt sugar, which you find in things like germinating grains. This one's a bit simpler. It's just 2 glucose units joined together. But check out the bond. It's an alpha-1,4 linkage, and unlike sucrose, look at the right side of this molecule. See that? One end has a free, available anomeric carbon. That end can actually pop open from its ring shape, exposing a reactive group. And that's exactly what lets it act as a reducing agent. And that, my friends, is why we call it a reducing sugar.
And our third key player is lactose, the sugar found in milk. It's made from a galactose and a glucose, connected by a beta-1,4 glycosidic bond. Just like maltose, lactose is also a reducing sugar because that glucose unit has a free anomeric carbon that can do its chemistry thing. Now, to digest this sugar, our bodies need an enzyme called lactase to break that specific bond. And if your body doesn't make enough of that enzyme, well, that's what leads to lactose intolerance.
So let's just recap that real quick. We've got 3 different pairs of sugars, joined by 3 different bonds, and they result in 3 totally distinct molecules. The big takeaway here is that everything matters. Which monomers you use, what kind of bond you make, alpha or beta, and which carbons you connect. All of that gives each disaccharide its own unique personality and properties.
All right, you with me? Good, because now we're going to scale up. If disaccharides are pairs, then polysaccharides are entire communities. These are the true heavyweights of the sugar world, polymers made of hundreds, sometimes even thousands of monosaccharides all chained together.
We can basically split these giants into 2 main camps. On one side, you have homopolysaccharides. These are pretty straightforward. They're built from just one type of monomer, repeated over and over again. Think of a long chain made of only one kind of link. Starch and cellulose are perfect examples. On the other side, you've got heteropolysaccharides, which are made from 2 or more different monomers, creating much more complex patterns.
Now, for the rest of this explainer, we're going to focus in on the homopolysaccharides because they have 2 major, and I mean really different jobs in nature, either storing energy for later or providing tough, rigid structural support. So let's start with the plant world's primary energy bank, starch. Starch is actually a mix of 2 different glucose polymers. On the left, we have amylose, which is just a simple, unbranched chain with alpha-1,4 bonds that naturally coils up into a helix. On the right, we have amylopectin. It's got that same alpha-1,4 backbone, but it also throws in some alpha-1,6 linkages.
These create branch points every 20 or 30 units, creating this awesome tree-like structure that's perfect for packing as much energy as possible into a small space.
Okay, so that's plants. But what about us animals? We need an energy reserve too, right? Well, ours is called glycogen. And as you can see, its structure looks a lot like amylopectin, but it's even more branched, with branches popping up every 8 to 12 glucose units. You can almost think of it like a dense little energy grenade. All those branches mean that when your body needs a quick shot of energy, say during a workout, enzymes can attack from tons of different points at once and release glucose incredibly fast.
And this is where it gets really cool. This is the perfect example of how one tiny change can change everything. Meet cellulose, the most abundant organic polymer on Earth. Just like starch, it's just a long chain of glucose. But here's the game changer. The link is a beta-1,4 glycosidic bond. This seemingly small flip prevents the chain from coiling up like starch does. Instead, it forms these long, straight, super rigid fibers. These fibers then line up and form hydrogen bonds with each other, creating the incredibly tough material that makes up plant cell walls. Our digestive enzymes are shaped to break alpha linkages, which is why we can digest starch, but cellulose just passes right through us. All because of that one bond.
Okay, so we've seen carbs as fuel tanks and as scaffolding. But now we're moving on to their most sophisticated role yet, acting as information carriers, specifically as the markers that give your cells their identity.
So how does a cell build this identity? It uses a team of highly specialized enzymes called glycosyltransferases. Think of these as the master architects of the carbohydrate world. Each one has a very specific job, to add a specific sugar to a specific molecule with a specific kind of bond, building these complex oligosaccharide chains one piece at a time.
And there is seriously no better example of their work than our own ABO blood types. On the surface of your red blood cells, you have these little oligosaccharide chains. Everybody starts with the same foundation, the O antigen you see on the left. Now, your blood type is determined by which glycosyltransferase enzyme you inherited from your parents. If you have the A enzyme, it tacks on a sugar called N-acetylgalactosamine. If you have the B enzyme, it adds a galactose instead. And if you have neither of those enzymes, well, you just stick with the basic foundation, and you're type O. It's that elegant and that important.
Now, these little sugar ID tags don't just float around by themselves. They're attached to other molecules, most often proteins. And when that happens, we call the whole thing a glycoprotein. These sugar chains can be attached in two main ways: N-linked, which is to the amino acid asparagine, or O-linked, to serine or threonine. And these glycoproteins are absolutely vital for everything from cell-to-cell recognition to signaling.
And here's something that's just wild. Even with all the incredible diversity you see in these chains, pretty much all N-linked oligosaccharides are built from the exact same starting block. As you can see here, they all share this core pentasaccharide that's a 5-sugar unit, made of 3 mannose and 2 N-acetylglucosamine units. From this one universal Lego piece, those glycosyltransferases build out the mind-boggling variety of structures that life depends on.
Okay, so let's do a quick flyby of what we've covered. We saw how simple sugars link up to form short oligosaccharide chains like sucrose and lactose. Then we scaled way up to the polysaccharides, and we found a crucial design rule. The bond type determines the function. Alpha linkages are for coiled, easy-access energy stores like starch and glycogen. Beta linkages are for straight, tough structural fibers like cellulose. And finally, we saw their most sophisticated job as information carriers that define our cellular identity, like our blood types.
So here's the bottom line. Carbohydrates are one of the most versatile and fundamental classes of molecules in all of biology. They are not just passive fuel sources to be burned. They are active participants, speaking a complex language of structure, energy, and identity that is absolutely essential for life as we know it.